Welcome, explorers, to the microscopic world of bacteria. These tiny organisms, invisible to the naked eye, are essential to life on Earth. From breaking down organic matter to aiding in digestion, bacteria play diverse and vital roles. But how do we differentiate these minute marvels? How do their structures contribute to their functions? Let's delve into the fascinating world of bacterial cell structures and uncover the secrets within. Understanding the differences between bacterial structures, particularly between gram-positive and gram-negative bacteria, is crucial in various fields, including medicine. This knowledge is fundamental for developing effective antibiotics, diagnosing infections, and understanding bacterial pathogenesis. So let's embark on this scientific adventure and explore the intricate world of bacterial cells. In the late 19th century, Danish scientist Hans Christian Gram revolutionized microbiology with a simple yet powerful staining technique. This technique, known as the Gram stain, differentiates bacteria into two major groups, gram-positive and gram-negative. The Gram stain involves a series of staining and washing steps. The difference in staining arises from the distinct cell wall structures of gram-positive and gram-negative bacteria. Gram-positive bacteria retain the crystal violet stain due to their thick peptidoglycan layer appearing blue or purple under a microscope. In contrast, gram-negative bacteria, with their thinner peptidoglycan layer sandwiched between two membranes, lose the initial stain and take up the counter stain, appearing pink or red. This fundamental distinction, revealed through a simple staining procedure, provides valuable insights into bacterial structure and function. Gram-positive bacteria are like formidable fortresses, encased within a thick, sturdy wall of peptidoglycan. This peptidoglycan layer, unique in its thickness, acts as a protective barrier, providing structural support and safeguarding the bacterium from osmotic stress. Embedded within this peptidoglycan fortress are teichoic acids, long negatively charged polymers that contribute to cell wall stability and play roles in various cellular processes. These acids act as anchors, binding to the peptidoglycan and reinforcing the cell wall's integrity. Gram-negative bacteria, unlike their gram-positive counterparts, possess a more complex cell envelope structure. Imagine a castle with not one, but two protective walls. Gram-negative bacteria have an inner membrane, similar to the plasma membrane of other cells, and an outer membrane unique to this bacterial group. Sandwiched between these two membranes lies a thin layer of peptidoglycan, offering some structural support but less robust than its gram-positive counterpart. The outer membrane, a defining characteristic of gram-negative bacteria, acts as an additional permeability barrier, controlling the entry and exit of substances. This outer membrane is studded with lipopolysaccharides, complex molecules with significant implications for bacterial pathogenesis. Section 5. Peptidoglycan, the bacterial chainmail. Peptidoglycan, the defining component of bacterial cell walls, is a marvel of molecular architecture. Imagine a chainmail suit providing both protection and flexibility to a warrior. Peptidoglycan, similarly, forms a mesh-like layered structure that encapsulates the bacterial cell, providing structural integrity and protecting it from osmotic lysis, a catastrophic event that can occur due to differences in solute concentration between the cell's interior and its surroundings. This chainmail consists of repeating units of two sugar molecules, N-acetylglucosamine, or NAG, and N-acetylmuramic acid, or NAM, linked together by peptide bridges. NAM, unique to bacterial cell walls, is the attachment point for these peptide chains, which cross-link the glycan strands, forming a strong interconnected network. Section 6. Size and shape, the bacterial fashion show. The bacterial world is a diverse and fascinating one, with bacteria exhibiting various shapes and sizes. From the spherical cocci to the rod-shaped bacilli and the spiral-shaped spirilla, bacteria showcase a wide array of morphologies. This diversity in shape and size is not merely a matter of aesthetics, it has significant implications for bacterial survival and function. The size and shape of a bacterium influence its surface area to volume ratio, which in turn affects nutrient uptake and waste removal. Smaller bacteria, with their larger surface area to volume ratio, are more efficient at nutrient acquisition and waste expulsion. Shape also plays a crucial role in bacterial motility. Some shapes, such as rods and spirals, facilitate movement through their environment, allowing bacteria to access nutrients, escape from harmful substances, or colonize new niches. Section 7. The fluid mosaic model. Membranes on the move. The cell membrane, a defining feature of all living cells, is not a static, rigid barrier, but rather a dynamic and fluid structure. This dynamism is best described by the fluid mosaic model, which revolutionized our understanding of membrane structure and function. Imagine a mosaic artwork with individual pieces coming together to create a larger, intricate picture. Similarly, the cell membrane, according to the fluid mosaic model, is composed of a phospholipid bilayer with various proteins embedded within it. These proteins, like the individual pieces of a mosaic, are not fixed in place but can move laterally within the membrane, contributing to its fluidity. This fluidity is crucial for various cellular processes, including transport, signaling, and cell division. Section 8. Feast or famine, bacterial life in two states. Bacteria, like all living organisms, are masters of adaptation, capable of thriving in diverse environments. They have evolved sophisticated mechanisms to cope with changing nutrient availability, switching between two distinct physiological states, the vegetative state and the starvation state. In the vegetative state, when nutrients are abundant, bacteria are metabolically active, growing and dividing rapidly. They synthesize essential cellular components, replicate their DNA, and produce energy to fuel these processes. However, when nutrients become scarce, bacteria transition into the starvation state. This state is characterized by reduced metabolic activity, a slowing down or cessation of growth, and the activation of stress response pathways. Section 9. Conclusion. The significance of tiny structures. The microscopic world of bacteria, though invisible to the naked eye, holds immense significance for life on Earth. From their roles in nutrient cycling and ecosystem functioning to their impact on human health, bacteria are essential players in the grand scheme of life. Understanding the structural intricacies of bacterial cells, particularly the differences between gram-positive and gram-negative bacteria, is crucial for various applications, including medical diagnostics and treatment. The Gram stain, a seemingly simple staining technique, provides valuable insights into bacterial cell wall structure, aiding in the identification and classification of bacteria. As we delve deeper into the microscopic realm, we gain a greater appreciation for the complexity and elegance of these tiny organisms. Their adaptability, resilience, and essential roles in various ecosystems highlight the interconnectedness of life on Earth and the importance of understanding and appreciating the microbial world around us.